Can I keep selling sex for money, officer? Citrate, isocitrate, ketoglutarate, succinyl-CoA, succinate, fumarate, malleate, oxaloacetate, joined in the Krebs cycle. Can I keep selling sex for money, officer? The Krebs cycle is a little bit understood by many people, but completely understood by nobody and understood very well by a select few. Let's look at 3 different functions of the Krebs cycle. First one being, when we take in food, how do we convert it into energy? That food might be carbohydrates, and carbohydrates are broken down, glycolysis, into glucose. Glucose is converted into a molecule called pyruvate. Don't worry about the structure here. And pyruvate is converted into acetyl-CoA. Acetyl-CoA is the fuel of the Krebs cycle when it burns that component and turns it into CO2 and energy. Another food is fat. Whereas glucose is turned very quickly into energy, fats are the most efficient way to generate energy from a source. Fats are turned by oxidative degradation into acetyl-CoA also. So we have 2 sources of acetyl-CoA to feed into the first intermediate in the catalytic cycle. Now, because it's a cycle, we can begin anywhere we want to. So let's take acetyl-CoA and combine it with oxaloacetate. That generates citrate. Citrate is just isomerate to isocitrate. Same chemical formula. Isocitrate is then partially decomposed with generation of CO2 and a molecule of NADH. That's a little pocket of energy used for reduction. We can call it reductive energy, if you like. And NADH is fed into the electron transfer complex. 4 enzymes on the surface of mitochondria which take NADH and other inputs and convert them into protons outside the membrane. Those protons accumulate, generate a potential, and finally that potential is suddenly released through an enzyme called ATP synthetase, a mechanical enzyme that works like a turbine and generates ATP from ADP. ATP is a reserve currency of the cell. It drives many aspects of biochemistry. So when isocitrate then is decomposed, it generates ketoglutarate. Ketoglutarate is then changed into succinyl-CoA and another molecule of NADH is produced. Succinyl-CoA is hydrolyzed to succinate, and in that process we generate an even stronger reductive energy source, FADH2. FADH2 feeds back into the energy transfer complex as well as NADH. The cycle goes on with conversion of succinate to fumarate. Fumarate changes into malate, and malate decomposes with liberation of another molecule of NADH into oxaloacetate, and the cycle is completed. This cycle is actually a cycle within cycle. Most of these reactions are enzyme catalyzed. They involve cyclic processes driving each step of the overall conversion of oxaloacetate all the way through back to malate and then oxaloacetate again. So we can look at the Krebs cycle in a more chemical way, and we can see all these steps that generate NADH and FADH2 to drive the energy transfer complex and other things in the cell. But the Krebs cycle can be adapted. If there's enough ATP, but not enough building blocks to generate biological molecules, well, the Krebs cycle can take acetyl-CoA and convert it to amino acids. The Krebs cycle can take acetyl-CoA and convert it back to fatty acids. for storage if necessary, or it can carry on and use other intermediates to form amino acids, some neurotransmitters, or heme proteins, including hemoglobin, which you're using right this minute to breathe. It can generate nucleic acids, and it can generate more or other amino acids at a different stage in the process. The cell is a master of balancing, homeostasis. The cell generates proteins from amino acids. It uses them, then breaks them down when they're in excess, they're not necessary anymore, to give amino acids again, which are reconstituted, sometimes other proteins, which are then used for a different function. The cell needs, obviously, nucleic acids to make DNA and RNA. The cell needs heme, and the cell needs sugars sometimes for emergency generation of energy in other places. So sugars are generated back from malate via a reverse process of glycolysis. In other words, reverse of breaking down a sugar is gluconeogenesis. How does the Krebs cycle appear to decide, should I generate energy or should I generate building blocks? Well, if intermediates in the Krebs cycle build up, they are excreted into the cytosol in some cases and send signals to the nuclei. The nucleus is the brain of the cell. It detects these epigenetic signals and therefore unwinds the DNA, transcribes and translates into certain proteins which are switched on. Those proteins serve functions in the cell, and when those functions are finished, other epigenetic signals will signal to the nucleus, stop, change your function, do this instead. So the Krebs cycle is at the very center of the metabolome. The Krebs cycle is feeding energy and intermediates to other processes in the cell, and it's signaling to the nucleus. I've said before in other videos that the Krebs cycle is at the heart of many health concerns in Western society today, including heart disease, cancer, neurodegenerative diseases, metabolic diseases, including diabetes, inflammation, even psychiatric disorders, and of course, aging. If there's too much energy going into the electron transfer complex, NADH and FADPH2 will build up. That sends epigenetic signals to the nucleus. Often, those signals are necessary, they're required. Sometimes, if a person keeps on taking in food, generating more NADH and FADH2, then this chronic backup of the electron transfer complex keeps sending signals to the nucleus, and they ultimately will cause mitochondria to die. Mitochondrial dysfunction is at the heart of the big health concerns of contemporary society. Don't mess with your Krebs cycle, because if you do, it'll mess with your mitochondria, and that is bad. I recommend this wonderful book called Transformer by Nick Lane. I put a link down below. It explains many of the concepts that I've talked about today. Thank you very much for listening. Bye-bye.